Hi everyone. Today we'll be looking at an introduction to genetics, focusing on terms and a few concepts you should remember from middle school. So starting out, I'm just going to show you a few terms that hopefully you recognize. You may not remember what all of them mean necessarily, but these terms will all be used in this unit. Genetics has a lot of its own jargon, so you need to learn these terms. So a few starting out: alleles, incomplete dominance. We'll talk about alleles more today. Mendel, hopefully you should know a few things about him. Monohybrid crosses, homozygous, dihybrid cross, heterozygous, genotype, Punnett square, phenotype, dominance, carrier, recessive, and pedigree, codominance, and sex-linked traits. Now you may not have seen especially codominance or sex-linked traits in middle school. However, for the vast majority of these you should have seen before. Alright, so starting off, what genetics is. Basically, genetics is the study of inheritance and variation in organisms. Genes control organisms' traits, and they do that by controlling what proteins organisms make. So when you have different traits from someone else, that's because your genes are slightly different, which means you make different proteins, and those different proteins cause your different traits. So different proteins cause different hair colors, eye colors, propensity to like certain things, all sorts of traits. Another term you need to know is diploid. Right? The prefix di means two, so that should give you a clue as to what diploid means. Diploid cells have two sets of chromosomes. Most cells in your body are diploid, and that means they have two genes for each trait because they have two chromosomes. So those genes can be alike or they can be different for each trait. So for example, if we're looking at plant height, maybe in peas, a gene for plant height might have a tall form and a short form. These different forms are called alleles. So an allele is a form or a version of a gene. Like you could have the allele for tall plant height or short plant height. Maybe you have the allele for yellow seed color or green seed color. Alright, looking at some of Mendel's laws. The first one is called the law of segregation. To segregate means to separate. So what this law says is that alleles get separated during meiosis. Meiosis is the process by which gametes or sex cells are formed. So this is the production of sperm and egg cells. Each gamete will only get one of the two alleles. Remember, we said you have two alleles for each trait, or for each gene, I should say. But when we make gametes, they have half as many chromosomes as our normal cells. So that means each gamete will only get one of those two. And thus, there's a 50-50 chance of which one it gets. So for example, if we were talking about these pea plants and their height allele, if a plant had one allele for tall, one allele for short, half of its gametes would have the tall allele and half of its gametes would have the short allele. But none of its gametes would have the tall and short allele, just one. And this is a diagram of that. So up here we have our chromosomes, and these little bands are showing the alleles. Okay? So during meiosis, these chromosomes separate, and now we have different alleles in these gametes. Next law is the law or principle of dominance. What dominance means in genetics is which one covers up the other. So we say that some alleles are dominant and others are recessive. The dominant allele is always expressed or seen. So if you have one copy of the dominant allele and one copy of the recessive allele, the organism is going to show the dominant trait. Right? So even if there's only one dominant allele, it still looks just like it has the dominant trait. We usually write this as a capital letter. It's usually the first letter of the dominant trait that controls what letter of the alphabet it is. So the recessive allele will only show up if there's no dominant allele present. Because if the dominant allele is there, it gets covered up. So you can only see the recessive if there is no dominant. So you need two recessive alleles to show the recessive trait. This is usually written as a lowercase letter. So another term that you will get very familiar with is genotype. This is the genetic makeup of an organism. This is the alleles of the organism for whatever trait you're looking at, written out as letters. So we use some terms to describe the genotype, like homozygous. The prefix homo means same. So homozygous means the same alleles. They're either both dominant or both recessive. So for example, if we're using this pea plant height allele, we might have a big T for the dominant allele for tall, and a little t for the short allele. We do not use an S for short. Use the same letter. Usually we pick the letter of the dominant trait. In this case, tall is dominant, so we picked T. So same alleles, and a plant could have big T, or it could have little t. So if there's homozygous for the same alleles, there's also heterozygous for different alleles. So in this case, it would be big T, little t, and it also shows the dominant trait. So this plant, big T, would be tall, and it would look exactly like this plant, big T. This plant isn't taller because it has two T's. Right? For now, we're dealing with complete dominance. So this plant looks the same as this plant. However, little t would be a short plant. So these are a few examples to test yourself. Big T represents a blank genotype. So in this blank, you need to say whether it's homozygous or heterozygous, and whether it's dominant or recessive. So big T represents a homozygous dominant genotype. That's what you should have there. Little t represents a homozygous recessive genotype. And then big T, that represents a heterozygous genotype. You don't have to say heterozygous dominant, because when you say heterozygous, that means there's one big allele and one small allele. So you don't need to say heterozygous dominant. It's just understood. Alright, we had genotype, which means we also have phenotype. Phenotype is the physical characteristic of an organism. So this is what it looks like. The genotype was the letters. Phenotype is the physical trait, so what it looks like. So tall or short in this example. One way to remember this is phenotype starts PH, just like physical. Phenotype, physical. So we said big T or big T shows the dominant trait, and little t shows the recessive trait. So in other words, big T, that would have a phenotype. Its phenotype would be tall. Jump over here, little t, its phenotype would be short. And now big T, is that tall or short? That one is tall because it has one copy of the dominant allele. Alright, so make sure you understand how to get those, and we'll move on. Alright, so dominant phenotypes. It is not possible to determine the genotype of an organism with a dominant phenotype just by looking at it. So in this example, if you have, now we're going to change up traits a little bit here. Now we'll go to flower color, and we have purple flowers and white. Now in these pea plants, purple is dominant to white. So if I show you a plant in purple, you don't know whether it's big P, which would be this, or whether it's big P. You can't tell just by looking. Alright, and so this is why they could be big P, big P, but they both look purple. Alright, last thing I want to touch on a little is Punnett squares. These are used to represent the traits of offspring based on the parents. So the basic Punnett square is called a monohybrid cross. The prefix mono means one, so this is only looking at a single trait. And we often use a few letters to represent things. We call the parents the P generation, and the first offspring is called the F1 generation. The second is the F2 generation. So you may see those terms. So here's a basic Punnett square with our big T, little t, and our big T, little t for both parents. So we have heterozygous. Right. So what we do to set up a Punnett square is you take the alleles of each parent, you write one parent on top, one parent on the side, it doesn't matter which parent goes where, and then this represents the separation of the gametes, or the alleles, during gamete formation. So this individual, which is big T, little t, it can only give a big T or a little t. So if we fill out our Punnett square, big T here, we will get something like this. Now you can see 1, 2, 3 have a big T, so that means 3 out of these 4 would be tall, 1 out of 4 short. Now this just represents probability in the offspring. So you can't say if there are 4 offspring, 3 are definitely tall and 1 is short, because that's not how it works. But each time they have an offspring, you'd expect a 3/4 chance of tall offspring, a 1/4 chance of short. And this cross is very similar, just with color. So I'm going to scoot through this a bit quickly. So this is showing same thing, but with the purple flower color. 3 out of 4 purple, 1 white. Percentage, there's 75% purple, 25% white. Now if you look at the different genotypes, we have 1 out of 4 is big P, 2 out of 4 are big P, and 1 out of 4 is little p. So we get a 25%, 50%, 25% for the genotype, which gives us a 1 to 2 to 1 ratio. We will come back to these ratios in the future. Now some traits that may or may not be exactly Mendelian or work like this in complete dominance. We know eye color now is not as simple as brown being dominant to blue eyes. Eye color in humans is actually controlled by multiple genes, so you can't do these crosses too easily with eye color. But some things that seem to be pretty simple: cleft chin, which is a division in the chin, widow's peak, which is when your hair goes down, like I have one, in the middle, hitchhiker's thumb, which I have a picture of here, tongue rolling, we're not too sure about. By tongue rolling, I mean forming a U with your tongue like this. But we're not 100% sure about that, and paper tasting we'll talk about. Alright, if you would like to look at some Punnett square practice, try these 2 problems on your own, and you can ask questions in class. Hope you enjoyed that.